Hello, everybody. In this lecture, we're going to talk about the intracellular structures of bacteria. So let's get right into it. By the end of this module, you should be able to define the term nucleoid, describe the function of prokaryotic ribosomes, and discuss how their structural features support the endosymbiotic theory, discuss the role of inclusion bodies and provide examples of them, and explain the basic structure and function of endospores and discuss why they present challenges in healthcare settings. Now, when we think about our prokaryotes, we have to keep in mind that unlike eukaryotes, they lack any membrane-bound organelles. However, they do still have some structure in there that's important. Now, outside the cell, we have our extracellular space, and inside we have our intracellular space. That intracellular space is going to be filled with cytoplasm, or also known as cytosol. Um, and inside of that cell, floating around in that cytosol, we have a couple of things of import, including some different inclusions. So we have the nucleoid space. This is where the prokaryotic DNA, which is usually a circular, well, as far as I know, they're all circular DNA molecules, and they're double-stranded, but they're just a single set of strands, like unlike where with like human chromosomes where we have two copies of the chromosome. And it gets bunched together. And so that's why when we're looking through at this TEM image, this transmission electron microscope, we can see that there's this kind of dense area here in red. This is the nucleoid region. Now, in addition to our nucleoid region, where we have our bacterial chromosome, bacteria can also have extra chromosomal DNA, and these are plasmids. The plasmids can come in, singles or multiples, and there are even some plasmids that cause cell death, but they are smaller than the chromosome itself, and they're also circular double-stranded DNA. They usually carry useful but not constitutive genes. So constitutive genes are genes that like have to be expressed. They're expressed all the time. They're necessary for um, the life of the cell. They also aren't going to even, you know, express other necessary genes, even ones that don't have to be turned on all the time. So they are not required for living, but they usually carry stuff that's useful. Big example would be resistance genes for things like metal or antibiotics. They may also carry special metabolism genes, and oftentimes, like in the case of Bacillus anthracis, they actually carry toxin genes. Now we have the ribosomes, and ribosomes in prokaryotes are similar to ribosomes in eukaryotes, in that they're made of RNA and protein. And the function of a prokaryote or of a ribosome is for it to make polypeptide chains. And so we'll talk more about this when we talk about translation, but it basically puts amino acids together. Now, when we're talking about prokaryotes, the big difference that you'll notice is the size. So the prokaryote ribosome is composed of two subunits, a 50S and a 30S subunit. And when combined together, they become 70S. Now that S is basically a sedimentation unit. It's like Feldberg unit or something like that. And it's used for measuring sedimentation on an ultracentrifuge. And so basically, where does it fall out in a tube when it's centrifuged down? And so when they add together, the 30 and the 50 don't equal an 80S subunit, they equal a 70S subunit because they're not additive. It really just depends on where it sediments. Now, one thing to keep that's interesting here is that the ribosomes inside of prokaryotic cells are the same kind of ribosomes that we'd find inside of a mitochondria or inside of chloroplasts. There is a rudimentary cytoskeleton that you can find inside of bacterial cells. Um, and it's going to have these long protein filaments that are going to help provide structure to the cell. And they can also be important for forming or for determining cell shape. For example, in your Vibrio species, you're going to have this um, protein, the crescetin, which is going to basically form a crescent that causes the cell to get that vibroid shape or that curved rod shape. Um, in addition to that, you have the FITZ ring, which is where division of the cell takes place. You don't have to know all these exact protein names. That's not something that I'm overly worried about you knowing. What I do want you to like recognize is just what is the function of the cytoskeleton in these, and knowing that they can control or contribute to shape. Now we have inclusion bodies. These are these little areas inside the cells. They're usually going to be some sort of insoluble product, and they can be bound to the membrane, but they don't have to be. Many of them are going to actually store things. For example, we have these magnetosomes. They accumulate magnetic iron, and they are common in organisms that are magnetotactic. These are organisms that can actually move towards or along a magnetic field. And then we have things like these carboxysomes, which contain carbon fixing enzymes. And so carbon fixation is what plants do, right? That's how we turn CO2 into um, That's how we turn CO2 into like sugar. So when a plant is performing photosynthesis, that is a type of carbon fixation. Now, in this case, we've got the carboxysomes, and these are found in bacteria, and they're going to be the that contain these proteins that do carbon fixation, including rubisco. Rubisco is the most abundant protein on planet, and it's a rate limiting protein for the Calvin cycle. The Calvin cycle, of course, is the carbon fixation cycle. Then we have endospores, and endospores are kind of this special case where they are a way that microbes deal with harsh conditions or dormant, going into a dormant state or quiescent state. And so what they're going to do is they're going to form these spores when they aren't going to be metabolically active. And when when will this happen? This is going to be in times of, say, metabolic stress. So there's no food around, or heat stress, or water stress, aquatic stress, freezing, radiation. Anytime that conditions are bad and your vegetative cells can't survive, they may sporulate or turn into spores, right? So just as a quick um, check on terminology, vegetative cells are cells that are actively growing, and then cells that are sporulating are turning into spores, and then spores, of course, are just the dormant um, protected cells. And so there are some clinically important spores. And for example, we have a couple of different organisms in the Bacillus genus, the Clostridium and Clostridioides genera. So this includes things like anthrax, which is Bacillus anthracis, or tetanus, which is Clostridium tetani, or botulism, which is another Clostridium. Then gas gangrene, so like really bad wound infections, that's another Clostridium. And then what used to be a Clostridium, but has now been moved over to, or at least used to be classified as Clostridium, but has now been moved over to a genus called Clostridioides, is Clostridioides difficile or C. diff. And of course, that causes severe diarrhea. And endospores can survive for an extended period of time on surfaces, even in healthcare facilities. And so, what I've got here is an image from the post offices that were attacked with anthrax spores back around 2001. It took upwards of 2+ years to decontaminate those facilities and millions of dollars before people could work in them again. And so that cycle of sporulation looks like something like this. So at the top, you have your cell, and when the conditions are bad, and if it's able to sporulate, it's going to start by copying its DNA, then packaging that DNA into, and then its needed stuff like ribosomes into its spore coat. So when we look here, we've got our cell, and it starts to engulf, right, where we kind of get this asymmetrical separation, where you get not a direct separation down the middle, you kind of get this like offset separation. Then you start to form the spore and then the cortex, and this is going to be some of the like layers inside. It's going to have a lot of the it's going to have some of the stuff necessary for when they go to germinate later. And then you're going to make layers upon layers upon layers, which help them become more heat resistant and chemical resistant. And finally, the spores are going to release, and that spore can exist for months or years or whatever. I mean, there have been they have found spores in like 1,000-year-old honey, I believe. And then finally, when the conditions are good again, the spore germinates and you get a vegetative cell again. So with that, you should now be able to define the term nucleoid, describe the function of prokaryotic ribosomes and how their structure relates to endosymbiotic theory, discuss the role of inclusion bodies and give examples, and explain the basic structure and function of endospores. So with that, I hope you enjoyed the lesson, and I'll see you soon. Bye-bye.